so today I'm going to tell you about our work sort of utilizing single cell and systems immunology analysis to look at COVID-19, both in adults and also in pediatric populations sort of towards the end. So a bit of clarification on what I mean by systems immunology. So we sort of do two kinds of it in the lab. One is what I call top-down systems immunology, with the premise that there's much that we don't know yet. So therefore, we need unbiased approaches to discover the components and also how these components interact and start to build what I call black box, for example, machine learning kind of predictive models of systems behavior. For example, why do some people respond one way versus another? So this is what I'm going to be focusing on mostly for COVID-19 here, because there's really a lot that we still don't know. The other kind is more bottom-up emergent phenomena systems immunology, with the premise that there's much that we can intuit. For example, when you look at a complex circuit diagram of cells and molecules. So in sort of this way of thinking, we aim to build more quantitative models of how these components interact and work together and give rise to emergent biological phenotypes. So, so in both areas, we sort of are obsessed with one, there's a common element, which is variation. So we study humans. So as you may well appreciate, there's a lot of variation in humans, both in terms of inter-individual differences, as well as how individuals vary over, for example, time before and after a perturbation. The other type that we obsess about is single cells, and really trying to understand, for example, single cell heterogeneity and whether we can start to link heterogeneity across these scales. The other aspect about variation is that in human in particular, because of the paucity of ethical perturbations, aside from a few, like vaccines, for example, we really need to leverage existing variation in the human population to learn new biology and start to ask questions about both correlations as well as whether something may be causal. And so leveraging that kind of variation in the human population is a unique aspect of studying humans. And I'll show you how sort of in our work, we attempt to link these two types of variations together. So just a very broad introduction of the kind of work we do. So a key question we often ask is, why do individuals mount variable immune responses? And can we start to predict such responses and uncover potential determinants so that we can start to modulate the response towards the desirable outcomes? So we also sort of conduct comparative analysis of the human immune states across a range of conditions. It could be healthy individuals, but also multiple diseases. And again, at the single cell level, beyond sort of cell type and cell subset differences, we're interested in understanding the function of cell-to-cell variations, even in very homogeneous sort of cell populations. So, in studying humans, this approach called CITE-seq has been the single cell profiling workhorse in the lab. So CITE-seq provides several modalities of information at the single cell level. It provides surface protein expression, the transcriptome, and if desired, we can also sequence the VDJ, so to get the sort of BCR, TCR sequences at the same time in the same single cells. And like flow cytometry, CITE-seq uses antibodies to measure protein expression. But instead of fluorophores, oligobarcodes are conjugated to these antibodies to allow the reading out of many proteins. So we work out sort of multiplexing approaches to enable high throughput data generation of multiple individuals over time. And so by barcoding the individual using both their natural SNPs, and we then also barcode the sort of the time component using these hashing antibodies, and more recently using these lipid-based barcodes. So together, this approach allows us to pool now in the range of about 40 samples and process all of them together. So for example, we can also pool all of them together and sort for specific populations to enrich for particular more rare populations and profile them at a single cell level, or antigen-specific populations in this way. So, so the general idea is we take, let's say from COVID patients, PBMCs, we, in many cases, actually sort, as I mentioned, for specific more rare population, but at the same time, we also profile the unsorted total. So in the current panel, we measure 180 surface proteins, as I mentioned, plus transcriptome and plus the VDJs. And then we sequence all of them together. On average, we get about 5,000 to 8,000 cells per individual sample in this way. So surface protein data in these single cells, they have unique protein specific as well as cell intrinsic sort of droplet-based noise sources. So we sort of work out ways to denoise and normalize, especially the protein expression data, which has received less attention from mRNA data, as you can imagine, because of the explosion of single-cell RNA-seq. So we have combined both experiments and computations to dissect these noise sources. On the right, I just give you an example. I don't have the time to go into the technical details of this particular approach, but you can see that looking at classical biaxial plots, after this kind of denoising and normalization, you can see much cleaner populations and less noise, and you can manually gate these types of data if you would like to. But of course, with a large number of proteins, we can perform unbiased clustering using the protein expression profiles to review both cell subsets as well as finer cell states. So one thing I want to emphasize is the protein really helps a lot, and being sort of in immunology, we are a bit biased and really initially always focus on the proteins first and see what kind of cell subsets and cluster it reveals. And we often find that that provides very informative and annotatable clusters because we can sort of use the field's knowledge on different immune cell subsets and markers to really look at the potential functional association of these clusters. And as you can see in this example here, that you can reliably get both the sort of the high level cell lineage subsets, and then you can further cluster them using just the proteins alone again, and really getting to these finer cell states, for example. So I just want to highlight sort of one aspect in thinking about this kind of analysis. What kind of variation do we see? So as I mentioned, there's the sort of the subject-to-subject variation. And then there's, for each individual in a given time point, you can sort of decompose the single cells into these cell populations within which you still have sort of single cell level information, and you can look at, for example, how a particular protein or phenotype may vary in that population of cells. And then there's the time component, which is critical in COVID-19, for example, such that you can sample the same individual or population over multiple time points. And that we can also have a perturbation, infection or vaccination, for example, and then have additional temporal information about the cohort. So based on this kind of information, you can start to infer or delineate one aspect of variation, which are these more stable individual subject characteristics. For example, could be driven by genetics, could be driven by certain environmentally induced states. And then we can start to build sort of statistical and machine learning models to decompose this kind of variation. So for example, we can ask, how does the expression level of a given gene depend on the where the cell population, the single cell may be coming from, the age and sex of the individual, and the time component. Is it coming from an early time point versus a late time point? And is it associated with a perturbation, for example, right? And then you can also model these individual subject effects. And then you can now look at the genes, for example, that are associated with the perturbation of interest. And you can start to look at, for example, specific pathways and within a particular population of cells. And so you can build ever more complex models and start to model these hierarchical levels of information and try to look at these effects and also try to infer dynamics. So the work I'm going to tell you in the next set of slides, it's going to focus on COVID-19, and really I want to emphasize that this took a village. So we're a part of a larger NIAID COVID-19 consortium organized together by Gigi and Helen Su. And the experimental data and analyses I'm going to present here were primarily led by Ken Liu, a graduate student in the lab, Andrew Martins, William Lau, both staff scientists, and also Nick Romaninoff, another grad student from the lab. So the four of them working together with the team really pushed very hard during the past year on COVID. And the COVID-19 samples I'm going to describe, they came from Brescia, Italy, from our colleagues. And this is really during the very early stages of the pandemic. And they were really did a fantastic job of collecting and getting the samples we need to do the analyses. And so the consortium together has put out quite a bit of information about COVID and including biomarkers, looking at biomarkers associated with outcomes, as well as, for example, revealing with Jean Laurent and Helen Su, revealing these antibodies against type 1 interferons in the very extremely severe individuals with COVID-19. But next, I'm going to tell you this work that was published a couple months ago from the lab, where we sort of look at the adult fairly critical COVID individual patients over the course of time. And these were all done using the kind of single cell approach I just mentioned with CITE-seq, and also together with other types of data, including circulating proteins and cytokines. And by integrating all of those together, we try to gain a better view of the dynamics of the disease course and the immune response, and how those are linked to outcomes. So in this particular study, we focus on 33 fairly severe, mostly severe and critical patients, and this is reflective of the early pandemic in Italy, and also with 14 matching healthy controls. And one of the early things we noticed when we sort of plotted, so we had data on their circulating proteins as well as a bunch of lab parameters like neutrophil to lymphocyte ratios and CRP. What we noticed was that even in the so each row here is a parameter, and each column is a parameter, each row is a patient. What we noticed was that while the most severe individuals or the milder patients we had, they are reasonably homogeneous based on these parameters. But when you look at this whole group of patients, they're all they're critical or ordinal score 6. They're very heterogeneous in terms of what their molecular profiles look like. Therefore, we thought that this categorical severity measure is insufficient to really delineate some of the individual differences. So then what we did is we built a machine learning model and asked, can we fish out a subset of these parameters such that at least within this training cohort, we can predict the different categories, right? So indeed, we found 8 such parameters. For example, the neutrophil to lymphocyte ratio, the lymphocyte count, IP10, and so on, and also IL-6 that can delineate these 4 categories, basically predict the ordinal scores really well. But what this provides, it's a more continuous score to quantify severity in this set of individuals, which you can see is very heterogeneous, but otherwise they would all have the same ordinal score. So we try to then validate this approach in an independent set of patients. So we basically apply the same formula that we learned based on those 8 parameters, apply it to an independent set of 67 patients. And we see that indeed in this independent set, we can again delineate the sort of ordinal categories. But again, you can see the heterogeneity based on this score in terms of the patients, even within a single category. The other interesting thing is that this particular score, we call it the DSM, it's also predictive of ICU admission status in these set of independent patients. So when you look at all the patients, the patient who got admitted to the ICU had an elevated DSM score. And I think the most interesting to me, it's this plot here, where even if you look at the patients who came in as ordinal score 6 or 7 or being labeled as critical, there's a clear delineation in the DSM score. So those who got admitted to the ICU, they had higher DSM scores than those who didn't. So that means this metric provides a continuous severity measure for allow us to have a bit more power to really look at the patient cohort as a whole here. So we then look at the single cell data that we generated from these patients over time. And we built a model that accounted for age and sex, and the timing is very important because a lot of the parameters we saw, they sort of vary over time. Type 1 interferon signature is a good example where, as you can see here, compared to healthy controls, despite the attenuation in some patients, you can see that compared to healthy people, all of the patients in general mount a robust, elevated type 1 interferon response in all of the cell types, basically. But when you look at the association with severity, we saw what others have also reported, which is that the more severe individuals, they tend to have lower type 1 interferons. And we had to account for time in order to make sure that's not because of the shifting dynamics, because type 1 interferon signatures, they drop fairly rapidly over time. So for example, if you look at even a mild individual, he or she may have a fairly elevated score up front in the beginning, but it drops fairly quickly afterwards, which makes sense. And then the other aspect I want to point out is the inflammatory signatures, which you can see that it's elevated, but then again, paradoxically, at these earlier time points, the severe patients, they tend to have lower inflammation to begin with in multiple cell types. So the other aspect we want to sort of uncover is what correlates with disease severity. And if you just take all of the variables in terms of gene expression states in specific cell types and ask which ones correlated with disease severity, you often end up with like a hairball like this, because lots of them, directly or indirectly, may be associated with severity. So what we did is we came up with an approach to try to statistically infer these more direct correlates. So these are basically correlates that even if you account for their correlation with the rest of the variables, they are still very strongly correlated with the severity. So DSM here is a severity measure, as I mentioned. So we found through this approach actually a small number of direct correlates, including a senescence or apoptosis signature in PDCs, and multiple states in the CD56 dim, CD16 high NK cells. So these all has something to do with either fatty acid metabolism signature or the status of inflammation. So I'm going to zoom in a little bit here. In that what we found is that the PDCs, they tend to exhibit a higher apoptosis signature in the most severe patients. So they're correlated directly with severity. So this fits well with the notion that with the data that if you look at circulating PDCs, their frequencies tend to be low, particularly in the most severe patients. We also look at others data to see whether there's any signs that the PDC may have migrated to, for example, the tissues. We didn't see, so far, we haven't found any evidence of that. So suggesting that apoptosis in these cells may be one of the reasons why these severe patients are making less type 1 interferons, because PDCs are a major producer. We also have other data on its correlation with circulating interferons, but I don't have the time to show you the details here. The other aspect it's intriguing is the fatty acid metabolism genes. They tend to be up in the more severe patients, while inflammation tend to be down. So these two categories, they reflect inflammation, as I mentioned. So when we look further, it looks like the status of the NK cells are driven by, potentially driven by IL-15. So there's a very strong correlation between IL-15 and disease severity. And IL-15, it's also associated with this fatty acid metabolism score in these NK cells. And like I said, despite this sort of persistent presence of IL-15 in circulation in these more severe patients, there's an attenuation in the inflammatory status of these cells. So it looks like these cells are perhaps in a bit more of a burnout state, where they have been exposed to this very heightened levels of IL-15 in circulation, while they mounted this somewhat strange metabolic signature, because both the catabolic and biosynthetic genes are up. But they also kind of downregulate these inflammatory signals, including interferon gamma mRNA. So the other aspect we noticed in the adults was this sort of flip around later in the disease course, around days 17 to 23 since symptom onset. So as I mentioned earlier on, there's this inflammatory attenuation. So I'm plotting you, for example, here the NF-kappa B genes as well as inflammatory genes. So in both the classical monocytes and the NK cells I just mentioned, they tend to be lower in the more severe patients. But once you sort of go past the 17 to, you sort of go into that 17 to 23 period, they start to become higher, or they stay higher in the more severe patients, while the milder patients are coming down. And you can see the same in when we look at circulating cytokines. So for example, in IL-13, IL-6, what you see is that in the more severe patients, they sort of stay higher or maybe even tick up a little bit, while in the more milder patient, they start to come down. So then we decided to sort of look at this further in an independent sort of set of patients for whom we have outcome information on. Some of them would recover from critical disease. So all of them came in as critical, and some of them would unfortunately die. So we wanted to see what happens in this two groups of patients during this critical period. Is there a divergence similar to the ones we saw based on the single cell gene expression profiles? And that's what we found. We found that if you look at specifically at this, we call the juncture period, we see that a number of cytokines, including IL-6, IP10, IL-17, they start to diverge between these two groups of patients. So you can see that, for example, IL-6 is shooting up right before this period in purple here. You can also see signs of tissue damage also going up. And for example, lipocalin is going up, and also endothelial damage is also going up in the critical and diseased patients. So it looks like there's a divergence going on. So here's just a quick summary and of what I've been telling you so far. So as I reported, there's this sort of early attenuation, but then later on shooting up of this inflammatory signature. So one question is, what's driving the early attenuation, and would sort of IL-15 make a good drug target? And then whether this is responsible for not containing this kind of potential second wave that we saw. And in fact, the second wave kind of phenomena has also been reported by Dunning and colleagues earlier in severe influenza patients, except that the timing was shifted maybe a week earlier. There they saw that around days 10 to 13, they start to see this kind of behavior. And so, yeah, so these are some of the questions. So next, I would like to sort of turn your attention to pediatric COVID-19 and also MIS-C. And here we have profiled a relatively small sort of pilot cohorts of these MIS-C subjects in pediatric, with the goal of also comparing them to the adult data we have. We basically use the same approach here to look at these individuals. So this project it's led by Ken Liu in the lab in collaboration with Octavia and Gigi's lab. And just a clarification here that these days in symptom onsets for COVID, it's the sort of the more acute, while the MIS-C, it's really days since their symptom onset post their COVID infection, when they come up with this kind of MIS-C-like symptoms. So when you look at the MIS-C patients versus pediatric healthy controls, we notice that, again, they have elevated interferon signatures and have very heightened inflammatory signatures, particularly in the in T cells, very strong activate T cell and B cell activation signals. And when you look at the pediatric COVID versus healthy, we see that compared to the adults, they have stronger, quantitatively speaking, stronger type 1 interferon responses. And perhaps this helps explain why they tend to do better also, and broadly speaking. And also they have these elevated signatures in antigen presentation across many of the cell types. And one thing in adult COVID, it's especially in the severe patients, is that antigen presentation pathways are severely down in DCs, but also in cell types like monocytes. So this, again, may explain why in kids, the response, it's overall, the outcomes are better. When we compare the MIS-C and pediatric COVID, what we notice is that the MIS-Cs, relatively speaking, have lower interferon signatures compared to the pediatric acute COVID cases. They also have lower antigen presentation signatures. And by the way, this may also be because of the fact that some of these folks are taking steroids in MIS-C, so therefore some of these pathways are suppressed because of the medication. So yeah, I already mentioned this point. So one last thing I want to highlight is sort of the B cells in MIS-C patients. We noticed that the level of memory B cells are significantly higher earlier in the disease course, and then they start to sort of lower. But taking this with a sort of a caution, because the numbers are relatively small. So when you cluster, further cluster sort of the B cells, it reviews sort of multiple subsets. And one of them, it looks quite interesting to us, is this cluster 8. It expresses high levels of CD11C on the surface. So all of these are proteins. And so, but it downregulates TRAP5 in terms of RNA, at lower CD21 and lower CD27 in this population. And this resembles cells previously described in lupus. They've described these extrafollicular B cells that generate autoantibodies that look like these cells. And if you look at this population, they are also more enriched in the MIS-C patients compared to COVID and healthy adults. So this is potentially an interesting population and subset of B cells to think about and whether they may be relevant. And if you look at the transcriptome in these cells and ask how they're different relative to other B cells, what we came up with is that they're highly enriched for a lot of the genes that's been annotated as relevant in autoimmune diseases, from lupus, one of the top ones, to multiple myeloma and other types of diseases. So finally, the other thing we notice in the B cells is that some of the specific cell subsets, particularly the memory and plasmablasts, they have a higher mutational frequency in the MIS-C patients. So I'm highlighting several clusters here. So these are the memory ones, 8, the 1, 2, 5, and 9, while the plasmablasts also. So if you look at the mutational frequency and looking at the VDJ frequency of mutations, you see that in COVID, they increase. But if you look at MIS-C, they are even higher compared to the COVID. Same thing in the plasmablasts. There's actually no difference between the COVID and the healthy controls in this case. Now, that could be also a function of timing. But when you look at the MIS-Cs, they're significantly elevated compared to the COVID plasmablasts. And if you try to correlate the cells at their phenotypes to mutational frequency, what you often find are markers of activation and memory. So you see that 27, it's positively associated, as well as CD71, which is a marker of activated memory, and so on. So this may point to sort of this kind of vicious loop where pediatric COVID has a more elevated robust interferon response than adults. And MIS-C patients also, as you saw, had very elevated type 1 interferon signatures. Though it looks maybe more attenuated than acute COVID, but that may be because of the function of the steroids. So these interferons, like an example such as lupus, may initiate sort of a self-perpetuating feedback loop to drive autoantibody production and activations of these B cells, which can feed back on the PDC themselves. And so, and as some of the earlier papers have shown, autoantibodies are highly prevalent in MIS-C patients. So whether the B cells I'm sort of discussing here may be potential drivers of those remains to be seen. But I think it's interesting hypothesis to look into. So with that, I just want to thank folks. I think I've thanked the folks along the way, especially folks at the Center for Human Immunology and my lab, and happy to take any questions.
Great, John, thank you so much. A lot of amazing scale. I can't even begin to think of the scale of the data that is being generated in these single cell surveys.
Yeah, awesome. Yeah. The good thing is, yeah, they're all out there, and folks can reanalyze and relook. That's the great thing. We can start to integrate with others. Yeah.
Well, one technical thing. So I'm not used to the ability to be looking at both RNA-seq data and protein data simultaneously, as you do. At one point, you mentioned that the protein profiling gave you better clustering or more clean clustering often than the RNA data. And I wondered if that was sort of generally true, and whether that's saying there's a lot of post-transcriptional stuff going on that's not, therefore the RNA doesn't reflect reality as well as the protein data.
Right. Yeah, so that statement, I have a bit of a bias in the sense that, as you alluded to, I think the proteins are definitely more stable, particularly the ones on the surface. And the other, there's of course a wealth of knowledge in immunology and other fields about markers that mark certain cell types and subsets. And so therefore, when you have hundreds of proteins on the surface, it makes the annotation easier. In the mRNA, often, if you look at for example, CD4 and CD8 is the good example. If you simply look at the RNA there, because they're so low and it can be noisy, that alone does not distinguish the CD4s and CD8s. And even if you use additional mRNA and try to cluster the CD4 and CD8s within the CD3 positive T cells, you can still see a lot of overlap. For example, between them, and the CD8 can sometimes be overlapping with the NKs. So, but when you look at the proteins, that's where, of course, because of what we know and also the stability of the markers, you can clearly separate them out nicely. So that's where that's why we kind of like to typically go in and look at the proteins first and use that to cluster the cells, and then we, within each one of the cell clusters, ask how do these transcriptomic differences differ, for example, across different groups or over time. So that way we sort of have a one level to anchor what the cells are, maybe and then look at their transcriptional states.
Right. And I suppose that reflects the limited depth you can do in single cell, RNA-seq. You've got to.
Yeah, both that and also naturally a lot of the mRNAs, depending on which one you're looking at, naturally they exist at very low levels in the cell. So even if you have perfect sensitivity, it could still be an issue. But having said that, because you have a lot of different transcripts in mRNA, the idea, of course, is to leverage that high dimensionality of information and use multiple RNAs together to define a cell. So often that also gives you very informative clusters. And colleagues, for example, Rahul in NYU has Satija their group have combined both types of information and tried to cluster cells that way. It also provides very informative results.
And the other terrifying thing is the timing, as you say, which has got to be so important because these things are changing rapidly, I'm sure, across time, and probably not identically in every patient, as the virus, either does or doesn't expand hugely. So, yeah. Exactly.
And yeah, well, that's one of the challenges is that within a single individual, it's typically quite challenging, of course, to collect to do very dense sampling. So therefore, sort of the one trick we rely on is to sort of bin these individuals into groups, for example, different shades of disease severity when they first came in. And then within each one of those groups, we try to model the effects of time by borrowing information across these subjects. So now time is reasonably covered densely. But of course, because of the inter-subject heterogeneity, some of which is biological and natural, they can basically both blur our resolution of what's going on, as well as hindering our ability to dissect the single individual outcomes.
Yeah, I guess, I can only do so many time points on a patient.
Right. And the most we have done are, of course, in other studies where we have healthy people, they came in, we give them a vaccine. So the most we have gotten is like something like 14 time points, but those are very different situations because if it's well controlled, right? We can. So for example, we wanted to study the innate response, therefore we sampled them like 6 times during the first 24 hours, but that's a much more controlled situation.